We are now at the half of the LIFESAVER Project. 2 years have passed already, where we have been able to integrate the different technologies into one solution.
We are in the era of digitalization, of computerization. We can sequence human DNA, we can predict diseases. Nowadays, we can also sequence the microbial DNA. The microbial DNA is so important not only for the gut dysbiosis, but it's also important and successful to fertilization. It is also very important to maintain a balanced microbiotic community in the vagina, and that would result in a likelihood of developing a success in fertilization.
I'm participating in the Consortium of LIFESAVER Project, providing, together with my team, contribution in the, I would say, in basic science. Because since more than 20 years, we are studying placenta, we are studying the different type of cells that we have within placenta, how these cells are communicating, which is the phenotype of these cells, how they are regulated, which are the main characteristics of these cells.
Our role in LIFESAVER is to add flow and microfluidic environmental control to the cells that are being developed by the other partners to model the placental interface. We can control the precise flow of fluid over cells in culture, and this is one of the major factors and stimuli that has been missing from cultures for the past decades. With this, we can improve the physiological relevance of the models, and we aim, together with the other partners and the expertise that we all bring together, to improve the placental model for risk assessment of chemicals.
We are specialized in making microfluidic systems, and in the framework of LIFESAVER we are actually building the platform to mimic the placental barrier.
Our core competence is the development of continuously growing human cell lines. We obtain tissue and isolate cells thereof, we cultivate them, and we try to turn them into continuously growing cell lines. We try to characterize them for the functionality that makes them fit for purpose for the LIFESAVER project.
My research expertise is bioprinting technology. This is a technology that allows us to stack human cells into 3D biological tissues. On the LIFESAVER project, we're using this technology to recreate elements of the human placenta and amniotic membranes. Bioprinting is really useful for this because these tissues are layered, containing multiple different cell types. We've made recent progress on recreating the amniotic membrane, and the next phase of the project will focus on recreating the placental membrane.
Main focus of research is mainly the use of these cells for therapeutical purpose. But here in this project, we are going to provide this knowledge about these cells as a big challenge to be able to build up in the best way the device that is the outcome of the LIFESAVER project, a device that will allow to study how different molecules, good or bad, can cross this structure that is the placenta and can reach the fetus.
We are a private bioprinting technology provider company, and we're also part of the LIFESAVER project as an industrial partner, supporting the project with 3D bioprinting technology, knowledge in bioprinting, but also the biomaterials used within the project. I believe the LIFESAVER project will contribute to advancing the way we do science, and more specifically what we do to support pregnant women and the fetus that is growing in the womb. Today, there are no in vitro models to understand how drugs and food additives affect the fetus.
This combination of disciplines makes it something exciting and new. So I'm really looking forward to what LIFESAVER is going to be in future.
The next 2 years, 24 months, we will try to get that closer as a solution to the people who need it. For that, we will be also approaching and collaborating with actors that are outside of our project with the aim of integrating all together more critical mass in order to get going our solution, and that everybody can take an advantage of it as a solution and as a future way of working together, technology meeting health needs.